Welcome to this final part of the video on valvular heart disease. In this video we're going to discuss prosthetic heart valves. So, prosthetic heart valves are the treatments for valvular heart disease, and they involve the diseased heart valve, which is either stenosed or regurgitating, being surgically removed and replaced with a prosthetic one. So, let's begin our discussion then of prosthetic heart valves. So prosthetic heart valves fundamentally come in two types. There are what we call tissue prosthetic heart valves, and then there are what we call mechanical prosthetic heart valves. Now, if you're having a tissue prosthetic heart valve, then that is a real heart valve. It's a heart valve that has been taken from some other being, either another human or an animal that has a similar sized heart to a human, and that has been treated with glutaraldehyde to fix it, and then is being transplanted into you. So tissue prosthetic heart valves are real heart valves, whereas mechanical prosthetic heart valves, those are not real heart valves. They've been made in a factory and they're being implanted into you. So those are the two different types of prosthetic heart valves: real ones, which are the tissue prosthetic heart valves, and mechanical ones, which are artificial prosthetic heart valves. So let's begin our discussion by going over the tissue prosthetic heart valves. So there are two different types then of tissue prosthetic heart valves. Either we can get the heart valve that we're going to implant in from another human, in which case the transplantation is what we would call an allograft transplantation. So I'll write this down here. So under tissue prosthetic heart valves there are two different forms. There's the allograft tissue prosthetic heart valve, which involves taking a heart valve, the equivalent heart valve, from a human who's passed away and then transplanting it into the recipient. Allo means same, so this refers to the fact that the transplantation is between the two members of the same species. So the donor is a human, the recipient is a human. Alternatively, the donor can be an animal, whether it likes it or not, and this is called a xenograft. So xeno refers to animal rather than it being the donor being a human. So this is where the donor is an animal, and of course then the recipient is a human. So animals that we can take heart valves from to implant into humans are pigs and cows, simply because their heart valves are around the same size as the human heart valves. So we take the heart valve from our donor, and then what you do is you fix that heart valve with glutaraldehyde. So let me write this molecule down. So glutaraldehyde, like so. And this molecule is very similar to formaldehyde. You've probably heard of the more famous formaldehyde before. Well, glutaraldehyde is going to do the same thing as formaldehyde. So let me just show you the structure of these two molecules. Formaldehyde is where you've got a single carbon double bound to an oxygen like so, and then with two hydrogens off it like so. So that's the structure of formaldehyde, whereas glutaraldehyde is effectively a longer version of that. Glutaraldehyde has 5 carbons, 1, 2, 3, 4, 5, and then at each end it has the same sort of structure. It has carbonyl groups with hydrogens coming off, like so. And this is the point where I wish I'd drawn a skeletal structure, because I now have to go through and put all the hydrogens laboriously on these intermediate carbons. So all the other carbons are just saturated with hydrogens like so. So this is the structure of glutaraldehyde versus formaldehyde. So you can see that effectively glutaraldehyde is just a bumped up version of formaldehyde. And they're both used to fix tissues. So what do we mean when we say tissue fixation? Well, this allows us to preserve tissues and prevent decay. So the reason these molecules are so capable of fixing tissues and preventing them from decaying is because they can cross-link proteins. You see, when you have a carbonyl group like this and a hydrogen also off that carbon, that hydrogen can come off, allowing the carbon to bind to some other molecule, And in the case of formaldehyde, you've got two hydrogens, so they can both come off and the carbon combines to two different molecules. And this allows formaldehyde to cross-link proteins. So if I just draw this here, when you soak a tissue in formaldehyde, what actually happens is if I draw two proteins here, let's say this is protein 1 and this is protein 2, you can get those two proteins cross-linked, like so, by the formaldehyde. So all you've got in between here is a carbonyl group, and that carbon of the carbonyl group is bound to functional groups on both protein 1 and functional group on both on protein 2 as well. Glutaraldehyde will do exactly the same thing. This carbon can lose a hydrogen and it can therefore bind to a protein, and this carbon can also lose its hydrogen and therefore it can bind to a protein. So glutaraldehyde cross-links proteins as well. So both of these, which are used for tissue fixation, the reason they fix tissues is because they cross-link proteins. And when you cross-link proteins in this way, it completely inactivates all of the proteins in the tissue, and that's really important for preventing the two stages of decay. So now let me just go over how tissues decay. So there are two parts to decay. One is called autolysis, and this involves the tissue breaking itself down. And the reason that that occurs is that when the cells in the tissue die, all of the lysosomes that contain loads of incredibly powerful enzymes capable of breaking down biological molecules, biomolecules, they are no longer under control. They break open and all the enzymes go everywhere and start breaking all the molecules down, breaking up the DNA, breaking up the proteins, breaking up the lipids. Everything is broken down, and that's called autolysis because the cell is basically breaking itself down because it's no longer under control of its own lysosomes. And remember, the function of the lysosomes in a healthy living cell is so that it can send anything that it wants to destroy there, and it will be broken down into its basic units, whether that's amino acids or nucleotides or whatever. Okay, so that's one of the stages of decay. So when a tissue dies, that's one of the reasons that the tissue will decay, because of cell autolysis. Fixing the tissue will stop this because all of the enzymes will be cross-linked and therefore they'll no longer function. So it prevents autolysis. The second stage of decay is given the incredible name putrefaction. And putrefaction refers to bacteria and fungal cells coming into the tissue, dividing in the tissue, and then gradually decaying the tissue, breaking it down so that they can use the molecules for their own purposes. So breaking down the human or animal proteins and lipids so that they can then use them for their own purposes. So this is the one where bacterial and fungal cells play an incredibly important role, so bacterial and fungal decay. And if you fix the tissue with formaldehyde and glutaraldehyde, bacteria and fungi cannot live in it. These two molecules are incredibly good at sterilizing tissue, so bacteria and fungi will not be able to live in tissue when it's being fixed with formaldehyde and glutaraldehyde. So this is the reason that fixation prevents tissue decay. So when you take a heart valve from either a deceased human or a deceased animal that you're going to transplant into another human, you firstly fix it because it's going to have to die, because you're not going to give it a blood supply in the new animal. It's just going to be inserted in to function mechanically. So you want to kill it, and of course tissue fixation will kill it. If any cells were left alive in the heart valve, they are now going to die. Okay, tissue fixation will fundamentally kill all of the cells, but it will mean that the tissue doesn't decay, and that doesn't matter. That is what's important. We don't care about whether there are actually cells alive in our tissue prosthetic heart valve that we've got. We just care about it functioning mechanically, and as long as the connective tissue doesn't decay, it's going to be able to do the function that we want it to do. So that's the process then. You fix your heart valve and then you implant it in, and then it will function mechanically exactly, hopefully, as it was doing in the initial donor. Okay, so that's tissue prosthetic heart valves. Let's now come to mechanical prosthetic heart valves. So there are multiple little designs for mechanical prosthetic heart valves, and I'm just going to go over three really important ones. So one of the designs is what's called the caged ball design, and this is where you have a little cage with a ball inside it. So let me show you this. So I'm just going to draw a little picture of one of these mechanical heart valves. So here at the base is the annulus, effectively. This is where the outlet will be for the blood to pass through, and then above this you're then going to have a cage like so. So here's one component of the cage, here's another component of the cage, and there'll be another bit going back like that. So you see what I've hopefully or tried to draw. It's this cage effectively, and I don't know how well I'm doing this. I'll try a little bit harder like so. So if we were to view this thing from above, what it would look like is like this. You've got a cross like that, and this is the cage. These bits coming up here. Maybe if I colour it in, it'll look slightly better. So I'll try colouring it in blue here. So this is part of the cage. This is part of the cage here. This is all part of the cage here. And then that bit back at the back there is also part of the cage. And then what you're going to do is you're going to put a ball inside here. So I'll get an even thicker pen. You're going to have a ball inside here, and it will be behind the cage. So now I'll replace the cage in here. So here's the cage back in front of the ball there. So you've got a little ball in there, and the idea is that the blood is going to be coming in this direction here. So let's say we've implanted this for the aortic valve. Let's say we're replacing the aortic valve. Then underneath, and I'll just get the thin pen back, underneath here is the left ventricle, and up here is the aorta. When the left ventricle contracts, pressure is going to build up there. It will push the ball up, and then it will open up gaps, whereas previously the ball was sitting in the annulus at the bottom here and therefore closing the valve. Whereas when it's pushed up into the cage here, then it will open up gaps. I hope you can understand that, and blood will be able to flow. And then when the pressure goes down in the left ventricle, the ball will fall back down and close the valve. So that's what we mean when we talk about the caged ball design of mechanical heart valve, and it's quite a nice simple design there. Now they're all really simple to understand, in fact. So next one is what we call the tilting disc design of prosthetic heart valve. And again, you're going to start off with an annulus like so, which will mark the outlet for the blood. And again, we'll just imagine we're replacing the aortic valve just for simplicity. And then in the gap, or in the middle of the annulus, what you're going to have is a disc that can tilt. So in the closed position, the disc will just be like this, but it's capable of tilting up, so it's capable of spinning upwards. And I'll draw it now in the tilted up position. So here, again, is the annulus. And now let's imagine that the disc is tilted up, and it's now in this position. So the disc has tilted, and you can see that it's now opened up the gap here. So again, imagining we've replaced the aortic valve, the left ventricle is below. When the left ventricular pressure goes up, it'll push the disc upwards here and open up the gap. When left ventricular pressure goes down, the pressure in the aorta will push this bit back down, and therefore the valve will close, is the idea. So it works effectively just like a normal heart valve. So that's the tilting disc design, and then the final design I want to go over is what's called the bileaflet design, and this really is just like a real heart valve. It's just got two leaflets, just like the mitral valve might have. So what you'll have again is an annulus, which will mark the outlet, and again we'll imagine that we're replacing the aortic valve just for simplicity. And then what you have is two leaflets, so one leaflet here and one leaflet here, and this is it in the closed position. When the left ventricular pressure goes up, it'll push both leaflets up, so both leaflets can flap upwards. So this one flaps up to this position, the other one can flap up to this position, so they've both flapped upwards to open up the outlet through which the blood can flow. When the left ventricular pressure goes down, the aortic pressure will push the leaflets back down and therefore close the valve. So that is the bileaflet design of a mechanical prosthetic heart valve. So those are the mechanical prosthetic heart valves. So we've now discussed both tissue prosthetic heart valves and mechanical prosthetic heart valves. If you have a prosthetic heart valve put in, you will need to be on permanent anticoagulant therapy, and this usually involves the very strong anticoagulant vitamin K antagonist warfarin. Now, warfarin works by stopping the liver from producing a whole bunch of coagulation factors, namely coagulation factor 2, coagulation factor 7, coagulation factor 9, and I'm now being tested on my Roman numerals, and coagulation factor 10. And, 2 is prothrombin itself. So, if you've stopped producing 2, you're not going to be able to initiate either of the intrinsic or extrinsic coagulation cascade, and these ones just make the block even stronger, not being able to have 10 and 9 and 7. 7 is involved in the extrinsic one, 9 is involved in the intrinsic one, I believe, and 10 is involved in both. So, stopping the production of all of these coagulation factors is going to lead to coagulation stopping, so you're not going to be able to produce fibrin meshworks from fibrinogen anymore. Without the fibrin meshwork, then you're not going to be able to form proper blood clots, because without that, the platelets don't really stick to each other properly. They'll stick to each other, but their interactions are extremely weak, and without that meshwork holding them together, it doesn't form a rigid blood clot at all. Okay? So warfarin very well stops the blood from clotting. And the reason you need to be on permanent anticoagulant therapy is because these prosthetic heart valves, whether they're tissue or mechanical, have a huge tendency to activate blood clot formation on them, and that's extremely dangerous. It could either cause pulmonary embolism if we're talking about a right-sided prosthetic heart valve, or potentially a stroke if we're talking about a left-sided prosthetic heart valve. So you need to be on permanent anticoagulant therapy after having a prosthetic heart valve installed. And if you've had a tissue prosthetic heart valve installed, you also need to be on a permanent immunosuppressant as well, to prevent the immune system attacking the tissue from the other being. So an immunosuppressant if you're on a tissue prosthetic heart valve. That's not important if you've got a mechanical prosthetic heart valve. So only if you've got a tissue one do you need to be on a permanent immunosuppressant, as you would if you had had any sort of tissue transplantation. Okay, and that completes the discussion of prosthetic heart valves. This is a very good treatment for valvular heart disease. It replaces the broken heart valve with a functioning one, and therefore will reverse all of the symptoms that came from having the diseased heart valve, such as the chest pain on exertion, and the lightheadedness on exertion, and of course also the heart failure process that follows valvular heart disease. So, thank you for watching this video. You've done very well to get to the end. I realise that it's many hours now since we begun. Thank you for watching it. I hope you've learned something.